Identical twins result from the fertilization of a single egg that splits into two embryos. They share the same genetic information, which means they have nearly identical DNA sequences. However, despite their genetic similarities, identical twins do not have the exact same fingerprints. The reason lies in the complex interplay between genetic and environmental factors during prenatal development. Fingerprints are formed during fetal development, specifically between the 10th and 16th weeks of gestation. The ridges and patterns on the fingertips, palms, and soles of the feet are determined by a combination of genetic factors and random environmental influences. While the genetic blueprint for fingerprint development is the same in identical twins, the actual formation of the ridges is influenced by various factors within the womb, including differences in the positioning of the twins, variations in blood flow and pressure, and other factors that are not fully understood. These subtle variations during the critical period of fingerprint development lead to the distinct patterns that differentiate one twin from another. Moreover, fingerprints continue to develop after birth and can be further influenced by the unique experiences and interactions each twin has with their environment. The friction and pressure exerted on the skin as twins touch objects and surfaces can modify the formation of their fingerprints over time, contributing to their divergence. In summary, despite sharing the same DNA, the formation of fingerprints is influenced by a combination of genetic and environmental factors during prenatal development and ongoing postnatal experiences, leading to the distinct patterns observed in identical twins.